Okay, next speaker is Professor Leo Poon, who's a professor and division head at School of Public Health at University of Hong Kong. Their group has been a pioneer in studying emergence, evolution, transmission of coronaviruses. They had major contribution in discovery and studying pathogenesis of original SARS-CoV, and again they are leading the field in trying to unravel the emergence and spread of SARS-CoV-2. So I'll request Professor Leo Poon. Okay, I see you, Leo. Hi, good afternoon, as in Hong Kong right now. Thank you for joining. I'll request you to share your screen.
Right, can you see it now?
Yes.
Right.
I'll put it live.
Okay, so maybe I switch off my camera. Okay.
Yeah.
So
just
now,
1 point, 5 minutes before the end of time, I'll just switch on my audio and remind you that we have 5 minutes left. Thank you.
Right. Okay. Thank you. So can I start now?
Yes, please.
Right. Okay. So first of all, thank you for inviting me to share our experience in the last 10 months about COVID-19. So it was actually, I mean, I agree with the previous speakers that we just live like almost 200% or 300% for the years. So because basically we have to deal with a new disease, and then we have been working so much harder than before to try to combat these emerging infections. Right. Okay. So first of all, maybe I spend a minute to introduce my faculties or my school. So I'm from School of Public Health, the University of Hong Kong. So our strength of this unit is tackling emerging infectious disease. So we have been handling H5N1, SARS, pandemic H1N1 in 2009, MERS, H7N9 in 2013, and so on and so forth. So we have been using multidisciplinary approach to try to use different models or different technologies to try to get to understand these pathogens, and in particular, how these pathogens be able to jump into human, so that we can develop evidence-based control measures to try to prevent the spreading of this disease to human communities. And we also work closely with various healthcare organizations like WHO or other international organizations like OIE and FAO. So we work together to try to combat these emerging infectious disease that jump from animal to humans, right? And then now, in the last 10 months, we have been working so hard to try to understand this new disease, COVID-19. So these are some of the publications or key publications that we had generated in the last 10 months. I do not think we have time to actually go through all of them, but then I think these publications are interesting. For those who are interested, please try to have a look, and they are all available in corresponding journal for free. Okay, so now go back to the COVID-19. So I personally heard about this disease on 31st of December 2019. Right, at that time, Wuhan Municipal Health Commissions basically had a press release to the public, and they mentioned about there was an outbreak of atypical pneumonia at Wuhan, Wuhan wet market, basically a seafood market. And they found a cluster of patients who had atypical pneumonia. Of course, retrospectively, we know that this is not the birthplace of this disease. We know that there were other COVID cases before this time, before December 31st, and also it happened elsewhere in Wuhan. But this is the first time that really I heard about this COVID-19. And then, of course, I mean, within a few days or 10 days, we already know about the genomes. And actually, this is a virus here. We call it SARS-CoV-2. At that time, I'm in the study group of this Coronaviridae Study Group. Basically, the committee to try to responsible to name these coronaviruses and in terms of nomenclatures. Right. So at that time, when we look at the sequence, basically we phylogenetically, it's very hard to distinguish these two, the SARS coronavirus to SARS-2, because they are very similar. And at that time, basically, we try to name this virus according to our standard algorithm. This virus should be classified into this species, severe acute respiratory syndrome related coronavirus. And then to distinguish this from the SARS-CoV-2, so we name this as a SARS-CoV-2 in that particular time. And of course, there are a lot of people talk about other ideas in naming the disease, but then naming the virus. But of course, based on our traditional algorithm, we have to name this virus as a SARS-CoV-2. And the disease that caused by this virus, we call it COVID-19, and this is the name by the WHO. Right. As I told you before, I work in the School of Public Health, so there are a lot of public health issues at that time. Even at that time, we don't know how this virus is going to spread. Or first of all, is it going to spread? Is it going to cause a pandemic? Is it going to just cause a small outbreak? At the time, we don't know, and there's a lot of questions that we have to answer. So the most obvious questions that I'm often asked by, like for example, news reporter. They ask about, do you know how this virus being spread? Is it by close contact, droplet, or by fomites? So at that time, we have already started to try to investigate these diseases so that we can get to understand the transmission mechanisms, and more importantly, we can able to identify the patient early. So what we did right at the beginning is try to develop a diagnostic test to detect COVID-19. Right. So I told you before, I heard about the news about this outbreak on 31st of December. And then within 10 days, China basically released the first genomes in the public domain. So we used 6 days to try to develop a protocol, to try to develop RT-PCR assay to detect this virus. And we shared this protocol to WHO, and WHO, within a few days, they released to the his WHO website and share the protocol to all the member countries. And on top of that, we also share our reagents, including India, in some of the labs in India, and then we share the reagents to them. So these dots represent the labs that have received our diagnostic reagents. Although this is in small scales, only a couple of hundred assays, but then it can help them to identify the first patients. For example, Egypt, Sri Lanka, and Nepal, and these countries actually use our assay to detect their first patients. And of course, this will be very helpful because they can able to slow down the spreading so that they can buy more time to develop additional control measure to prevent the disease spreading in their community. But with this assay, we can do so much more. First of all, we managed to use this assay to quantify the amount of viral RNA in COVID-19 patients. Right. The chart right here basically shows that the viral load of COVID-19 patient from day -1 to day 15. Okay. So what you can see that here is you can really tell that actually COVID-19 is very infectious because right at the beginning, within the first 10 days of disease onset, you can detect high amount of viral RNA in both fluid or sputum samples, right? And then these viral RNA start to subside in the second week of disease onset. So by that time, so right at within the first week of disease onset, they are very infectious. Not only that, look at this, day -1. So that is the time the virus is still actually shedding a lot by the patients before they actually have any symptoms. So that is the real problem, because the patient without any illness, and they can actually feel free to go into the community, and they would able to spread the disease to other individuals, like in the market or in the office, and so on and so forth, right? So so that is actually very dangerous in compared with the situation in SARS coronavirus in 2003. At that time, what we found was, at the beginning, within the first week of disease onset, the viral load of SARS patients actually is relatively lower than what we can find in the second week of disease onset. So at that time, when the patient is getting very sick, they have been hospitalized, and so we can able to contain the disease and prevent this patient to get into the community at the time that they are most infectious. Right. So with these findings, we already immediately actually write it in, make a comment in my commentary, our commentary in these journals. So basically, we just warn the community that, hey, the control measure that we use for controlling SARS would not be very useful in controlling COVID-19 because the disease or the virus starting patterns is quite different from what we see in SARS time. So now, why the patients is so infectious in the first place, right? So what we did and during that time is I tried to use different tissues, human tissues, like bronchus, lungs, and conjunctiva, and infect them with different viruses like SARS-2 and SARS coronavirus in 2003. So you can tell that compare this one and this one. This is the bronchus. You can see that those show up in brown. These are infectious. Infected by SARS-CoV-2, right? And this one is infected by SARS. You can tell that, oh, this virus SARS-2 actually is very infectious. It can replicate very well in the bronchus, in the lung, in the conjunctiva. By contrast, SARS, coronavirus, it didn't go very well in the bronchus, and it cannot infect conjunctiva, right? So when we translate this observation into this chart, basically this is TCID 50 of this culture. You can see, compare this and the green chart, green bars and the orange bar. You can tell that the COVID-19, so SARS-CoV-2 actually replicate very well in bronchus, lung, and conjunctiva. By contrast, the titer coming from tissues infected by SARS coronavirus in 2003, it didn't actually grow very well in bronchus and not in conjunctiva, right? So what we can control right here is actually the virus is actually very fit. It can replicate very well in all different type of tissues from humans. And not only that, if you look at the amount of cytokines or chemokines released by the infected cells, you can look at here. So this is a control, which is a MERS virus. Tissue infected by MERS, coronavirus. You can see they actually stimulate very high level of TNF alpha, IP10, and aromatase. And when you look at the SARS-CoV-2, so there's not much there. So on the one hand, what we can control from here is, on the one hand, the virus actually grow very efficiently in human tissues. On the other hand, is even it has been able to infect the tissue, it failed to trigger a good innate immunity in the infected tissues, so that the human tissues cannot able to react to the infection and control the coronavirus replication inside ourselves. So that may explain why the virus is so fit right at the beginning of the disease onset. It can grow very rapidly at the first few days of the disease onset. Right, okay, so and then the other question is that we would like to answer is how this virus being transmitted between humans, right? Could it be by contact transmission, aerosol, or formal transmissions? Again, what we learned from SARS in the past was hamster actually is a good model for studying SARS. So my colleagues, Dr. Yan and her team, managed to use this hamster to develop a transmission model. So what she did was she tried to put a naive hamster together with an infected hamster, and you can see that the infected hamster actually have been able to replicate, support virus replication very well. But then also for the naive hamster, only 3 days after disease onset, those contact, you can already detect a high titer of viruses in the naive hamster. So contact transmission is possible. And then she did another experiment, is try to put 2 animals in the same cage, infected one and naive one in the same cage, but then putting wires separate them too, right? And so that this naive hamster to be infected, that can demonstrate droplet or aerosol transmission is possible. And this is what exactly she found. Again, the naive hamster could be shown to be infected 3 days after post-exposure, right? Actually 3 out of 3 could get infected at the end. So these two transmission mechanism is possible for COVID-19 in hamster. And how about format? Right, so she also managed to demonstrate this. So what she did is to put a hamster into a soiled cage. So a cage that has been used by an infected hamster, right? And then you can hear, so here, I mean, 1 in 3 naive hamster got infected, and it shed a high titer of CIDR coronavirus 2 virus in this particular experiment. So this free transmission model are possible for COVID-19, suggesting that this could be the same case for human. Do remember that this study has been done very early at the beginning. So at that time, we are not completely sure which is the major transmission mode for COVID-19 in human. Right, so if these are showing that COVID-19 is so infectious, this is not surprising that we can find cluster in restaurant, in household, and so on and so forth. But in this slide and the next slide, I'm going to show you some very interesting scenario which we found in Hong Kong. So this is the first example showing in-flight transmission of COVID-19 is possible. We actually have, is the first case, so we have the genetic evidence to support this claim. So this case, this particular case occurred in March. There was a flight from Boston to Hong Kong. In this particular flight, there were a couple that has been infected by COVID-19 at that time. They were in incubation period at that time. So here, so the flight was moving from Boston to Hong Kong on 9th of March. At that time, the patient did not feel anything. So they are actually asymptomatic at that time. But then when during the travel, they develop symptoms. They have cough and so on and so forth. And then they get back to Hong Kong, and then they have been detected by positive for COVID-19 subsequently. And in this particular flight, we also identified 2 additional cases, case 2, C and D. These 2 were found to be positive about 10 days after the flight. All right, and this one of them, case C, we managed to find them because of active contact tracings. We actually tried to trace those people who stay in this cabin. And then we managed to find case C. And then case D, she actually felt ill after the flight. And then she went to the hospital. And then she was test positive subsequently. So these 2 cases, case C and case D, actually they have cabin crew. All right, so they are not passengers. So it is unlikely for these two group of people mingle together before they get onto the on the flight. So, and then on top of that, we managed to sequence these viruses, and then these 4 viruses actually identical, and they all belong to clade G. At that time in Hong Kong, we don't have these 4 clade G viruses in Hong Kong. So these demonstrated that in-flight transmission is possible, and it is not exceedingly rare. So respiratory protective precautions should be taken when they get onto the flight. Right, the other interesting transmission is a event from pet owner to a dog. Actually, we found it twice in that particular report here. Right, and then again, we found there was COVID patients on 12th of February, and then she was detected positive, and then she needed to be hospitalized. And then she also found to infect another household member, and she also needed to be hospitalized. And with this situation, the pet has to be actually being looked after by the government, by Agriculture Department in Hong Kong. And then we managed to actually test this particular dog. And a few days after, right, actually on the same day, that this dog being sent to the quarantine, and we were managed to test this dog, and then found that this dog is positive for SARS-CoV-2, right? And then we also found a similar scenario in March. Again, there was a patient, and she need to get into the hospital, and we test the dog again from the owner too, and this dog also found to be positive. And on top of that, we also able to bled this dog, and the dog also found to be seroconverted, right? And then we look at the sequence. basically these two samples clustered together and these two samples clustered together, suggesting that COVID-19 could be transmitted from human to pets, right? And then without telling you the details, and we also managed to find there was cases from human to cats in Hong Kong. Right. Now, I told you before that actually these patients, COVID-19 patients, are very infectious. They're shedding a lot of viral RNA right at the beginning. But then when we actually try to stratify these patients according to their disease severity, in this particular study, stratified them to mild cases to severe cases. And what we found was for those severe cases, they tend to have a higher viral load, right? The orange dot here. And for those mild cases, they tend to have less viral load. So this is the delta CT. So the more negative it is, the higher the viral load they are, right? You can see that from within the first week of disease onset, the viral loads for severe cases is higher than the mild cases. And similar observation can be found in second week and beyond, okay? And not only that, those mild cases seems to shed less time than the severe cases. The severe cases, actually, we can tell that they shed a much longer period of time. Day 225 in this study, but then we have study here. We found some of the patients actually can shed viral RNA up to 70 days. Okay. The good news of this is we know that these samples collected from day 10 or beyond, they don't have infectious viruses there. So even though they contain a lot of viral RNA, they are not infectious. We try to isolate them by using cell lines, but none of these samples in this box is found to be containing infectious virus. So the only infectious period of the COVID-19 patients, most of the time will be up to day 10. So these are the most infectious period for COVID-19 patients. So here, so in Hong Kong, we try to isolate these patients for 10 days, and thereafter we try to bleed them to see whether they have been seroconverted. Alternatively, and see we find them whether they actually start to stop shedding viral RNA in the patient. If they have fulfilled any one of these criteria, we can send them, release them from the hospital. Right. Okay. Talking about antibodies. Again, we try to compare those antibody response from severe and mild illness, right? Those severe cases in here are highlighted in pink, and the mild cases highlighted in blue. So these are different time points, right, from first week of disease onset and to up to 60 days to 65 days here, all right? And you can immediately tell that for the severe cases, they tend to have a much higher antibody titer. This is the antibody titer against the RPD domain of spike, right? And we measured it by ELISA. And so you can see that even in the second week of disease onset, it is already very high and then maintained throughout the period of time. By contrast, the mild cases seems to have much slower kinetics. So for the first week is low, and the second week, yeah, we can start to detect it, but then it's almost 10 times less than the severe cases. And not only that, when you look at the chart here, and we always found there were some mild cases which are seronegative in the assay, suggesting that some of these COVID-19 patients, in particular mild cases, they may not able to mount a good antibody response against COVID-19. Right, and we also try to do another study using another assay. We use neutralizing antibody assays. So neutralizing assays against the COVID-19. Again, we try to stratify them according to the disease severity, severe, mild, and asymptomatics. You can see that it is much similar to what we found here, right? The severe case tend to have a higher antibody titer after the infections, whereas the asymptomatic one, they seem to have a much less antibody titer. Neutralizing antibody titer. Here is about 1 in 20, and this is 1 to 160. All right, and on top, and also in addition, we found the durations of this antibody titer for the severe case seems to be much longer than the asymptomatic one. So the red line is basically the detection limit of our assays. And we've estimated that it takes about 182 days for the severe patient to have the antibody titer drop to the level below our detection limit. Whereas for asymptomatic cases, right, the virus the antibody titer only lasts for about 90 days. After 90 days, after 90 days of infections, the antibody titer actually will fall below the detection limit of our assay, right? For the intermediate one, the mild cases, it takes about 150 days. All right, so you can see there's a different level of antibody response, both neutralizing antibodies and also the antibody against the RPD domain of spike. Right now, that is a problem. Now, if such a rapid decline of this antibody titer in our body, what is suggested that we may be infected by COVID-19 again after a certain period of time. Indeed, this is the case. But this is the first case of COVID-19 reinfection that detected in the world. This is happening in Hong Kong, where at that time, actually only a couple months ago, 2 months ago. So we've identified a patient that has been infected by COVID-19 twice, right? The first incident of this infection was found in March, and this patient is actually a Caucasian, 33 years old male, and he was infected by COVID-19 in Hong Kong, right? And then he was discharged. Basically, he was fully recovered and discharged in April. And this patient has been tested consecutively negative by RT-PCR assay, right? So this fulfilled the discharge criteria, but the problem of this patient was actually this patient mounted a very weak immune response against COVID-19. This is the neutralization antibody titer of this patient. You can see that up to day 43, we do not manage to detect any neutralizing antibody from these patients, right? And then for the ELISA assay for the RPD domain of IgG of spike. So again, it's only weak positive, right? And no matter what, this patient was negative consecutively by RT-PCR assay, so she was discharged. And he went to Spain in this summer and then come back at the airport, right? At that time, the Hong Kong airport actually had a mandatory testing for COVID-19, so he has to supply his sample to the government. And surprisingly, this patient was test positive again, right? And at that time, it was about 148 days after the first infection, okay? So, and the CT values is 20-something, so it's actually reasonably high. It's unlikely that the patient can keep on shedding this virus for that much period of time. And to just make sure, actually, we sequence both samples, causing the first infection and the second infections, right? You can see that genetically, these two infections are caused by two different strains, right? One from B2 and the other is coming from B1, okay, lineage. So confirming that this patient has been infected twice. And now with this serological data, that really suggesting that the infection of COVID-19 is possible. And that may have some implication, like, for example, vaccines, herd immunity, and so on and so forth. So we really have to think about this particular problem seriously. Right, and now talking about serological assays, we also recently managed to find out that actually our infection, COVID-19 infection, could trigger a good antibody titer against the ORF3B and ORF8. So these are the titer that we can manage to detect against ORF3B and ORF8. So, and we also know that the patient actually have a good antibody response against the N protein as well. So these three proteins, N, H, and ORF3B, are good serological marker for making or confirming a diagnosis, right? So in fact, we actually try to use these three antigens as a antigens to try to make some serological detections, And we actually have some good response. And this paper has been published in Nature Immunology for those who are interested. You may have a look on this. The good thing of this RF3B and 8 are because these two proteins is unique. They are unique. Even SARS coronavirus have this protein, but they are the protein sequence of these two proteins are completely different from those found in this virus. So there will be less risk of having cross-reactivity, which may help us to make a more accurate diagnosis. Right, now I told you before, formal transmission is possible, and a hamster who's put it in a used cage or swallowed cage could be infected. Right, and then I also being asked by reporters, like for example, if I'm sure you heard about this Diamond Princess scenario. At that time, there was a super spreading event in this particular ship, and a lot of people get infected. And I often ask by the reporter, hey, if we have a situation like this in Hong Kong, how we should do the decontamination, right? Should we wear a full PPE to get into this ship to try to do surface cleaning, or we should just wait for a certain period of time so that we're hoping that the virus will be inactivated by itself after a certain period. So, and in fact, we did an experiment in a BSL-3 lab. We tried to add different SARS-CoV-2 virus into different tissue objects, like put it on a tissue papers, glasses, and surgical masks, and things like that. And we found that this virus actually have a different half-life in different surfaces. Like, for example, if you put it on tissues, papers, it only lasts for 3 hours. And then after that, you will not able to detect any infectious virus thereafter. However, if you put it on stainless steels, this virus can survive from 7 days, right? So the virus actually can have a different stability on these surfaces. And that may have an implication for us to try to determine what we should do after finding a super spreading event. In fact, for example, like CDC in US, they make recommendations. Say if someone actually contaminated an area or like a room, you can seal the room for 7 days. After that, you may just do a very simple surface decontamination. You have a very little risk in getting COVID-19 because of get into this room after 7 days. The other good thing is we also found that the virus is actually very vulnerable to very simple detergents like bleach, ethanol, polyvinyl iodides. These are effective. If you just put them, mix it with the virus for a few minutes, you can inactivate the virus easily. Right, and then we also found the virus actually very sensitive to temperature. The virus can be inactivated easily, like at 70 degrees. And if you incubate the virus like the culture in VTM for 5 minutes, the infectious titer drop to below the detection limit. Right. In fact, we also try to develop some protocol to try to inactivate used masks. Do remember that at that time they're running out of surgical masks, running out of the N95 masks in hospital setting. So we developed a protocol like putting these used N95 masks, put it in an oven, a dry oven, for 60 minutes at 60 degrees Celsius. And we found that this will be a very effective inactivate disinfection procedures to try to inactivate the SARS-CoV-2. And we know that this protocol can able to recycle the used mask for 10 times. And this is the publication for those who are interested. Please have a look. And now, we know that the virus actually is very sensitive to temperature. And how about humidity, right? And we did a series of experiments. We put the virus on different surfaces like masks, plastics, stainless steel, and wood. What we found was the virus actually is very sensitive to temperature at high relative humidity. So I just use this example. This is the virus put on a stainless steel, and we put them in the same humidity, high humidity, relative humidity up to 95 degrees percent. And then we incubate it at 33 and 22 degrees Celsius. You can see that at 33 degrees Celsius, the virus can be inactivated quite rapidly. Whereas if you put it at 22 degrees Celsius, the half-life is a lot longer than these conditions, right? And then if you put these items on at a low relative humidity, the temperature effect is less obvious, okay? You can see that both temperature, 33 degrees Celsius and 22 degrees Celsius, it won't affect the stability of this virus that much, right? They're quite stable at low relative humidity, right? And then if you put them into a condition with a low temperature, low relative humidity, and a high temperature and high relative humidity, you can see the maximal differential effect. Okay, so this is the condition with high temperature, high relative humidity, and this is the condition with low temperature and low humidity. So that may also provide some additional information for us to try to inform us how to do a surface decontaminations for a contaminated area or contaminated items.
Leo, we have 5 minutes.
Right. Okay. It's about the end. Right. Now. Right. Now this is the situation of COVID-19 in India, in Hong Kong. I told you before, the virus doesn't like a high temperature, high humidity. So now these two peaks actually contradict what we observe, right? This is the summertime or monsoon seasons in India, and this is the summertime in Hong Kong. We have COVID-19 peaks in this time point. So that really reflected that COVID-19 transmission may not primarily caused by formal transmission, but human behavior like close contact or droplet transmissions is maybe the major mode for human-to-human transmission. So that's why, as a public health practitioner, we always emphasize people to maintain a good hygiene and wearing masks, social distancing. This is found to be very effective in reducing the spreading of the disease. Although maintaining good hygiene is also good, but then if you look at the peak here, really these two transmission modes maybe is primary factors that account for the spreading in these countries. Right now, I'll talk about these formal transmissions, and we also did some experiment to try to develop we call smart surfaces, so that we can put some coating. Like this is a doorknob. We try to coat this doorknob with cuprous oxide, and we put it there. And what we found is it can able to reduce the viral infectivity by 3 logs just within an hour. So, example, if you coat it in a stainless steel, so it could drop from here to here. This is without coating. This is with the coating. And this is the same case for coated glass. This is without coating. This is with a coating. So you can see there's a dramatic reduction in the titer. So we found that cuprous oxide may able to inactivate the virus. We are now still working on the mechanism to try to understand how these metals can able to inactivate the virus. Right. Okay, so this is my summary. So I have told you basically how these COVID-19 patients shed the viral RNA during the different time points. So basically, it peaks at the first peak of disease onset. And I also talk about the serological response of COVID-19 patients, right? And then I also explain the transmission mode and the zoonotic transmission of COVID-19 in Hong Kong. And I also briefly mentioned about the virus stability and different environmental conditions, right? But do remember that this may not be the only incident that we will encounter. And then, in fact, there are a lot of many different viruses from bats. It may able to jump into human in future. We know that SARS was coming from bats. We know that SARS-CoV-2 may be coming from bats. And there are many examples like OC229E. We know that it may become from bat as well. So many coronavirus in bats still have plenty of opportunity to jump into human in future. So the challenge that we have is how we can prevent this spillover event in future, right? And right now we are experiencing COVID-19. We know that the virus can jump into pet, and recently it can also it was reported that it can jump into mink as well. And then the bad news is there may be some evidence to suggest that some of this mink virus can jump back to human and infect human. So maybe the gene pool of this virus may be much bigger than we've appreciated in the past. So we need to try to come up with a proper control measures to reduce the spreading of this disease in future. Right, at the end, I would like to acknowledge my colleagues from School of Public Health and other departments under Hong Kong U, and then various departments from Hong Kong governments and overseas institutes. I would like to thank all the colleagues in other healthcare organizations and also the funding support from these agents. Right, at the end, I would like to thank for your attentions. Thank you.
Thanks a lot, Leo. You can stop sharing the link.
Yep. Okay, and just a minute.
Okay.
Right. Good.
Yeah. Really amazing body of work done in such short period of time. Thank you for all the effort on the people in Hong Kong U. We'll just take a few very important questions. One is about reinfection scenario where you looked at if someone has an asymptomatic encounter, the antibody longevity, not really longevity, the titers will drop pretty soon, and there's chance of reinfection. Have you looked at the memory response instead of just titers?
We just managed to get the titer work done. So we would like to, we'll continue to work on this, studying this case, but then, you know, science takes time. Unfortunately, I cannot give you an answer at the moment. But I would believe this is not a unique event. Because if you look at the chart that I showed you before, there are actually quite a few people who do not seroconverted after the infections. So I will predict this is not uncommon. There will be other individuals will be infected again. Bear in mind that this patient that we were able to detect it because of the mandatory testing at the airport, right? This patient actually is asymptomatic at that time. He even don't know he was infected, right? And he was perfectly normal. So without this mandatory testing, we were not able to define him. So this depends on how much effort that we want to look at in this population.
All right. Okay. Second question is about the shedding of RNA at early stages. Shedding of RNA, does it really correlate with shedding of infectious virus also, or?
Not really. I mean, in the first week, in the first days of disease onset, yes, there's a correlation. The higher the titer, the more likely you will manage to isolate the virus. But then after 10 days, first of all, we failed to isolate any infectious virus. But we know that there were some patients actually shedding a lot of viral RNA, CT like up to 30, 20 something. We have such an incidence in the past. So and then they may actually shed the RNA up to day 70. So yeah, we don't know why, but this is not unique. That has been reported in Europe, US, and many other countries as well. We don't know the reason why, but this is something that we need to pay attention to.
Correct. Okay, thank you so much. A lot of other questions were self-explanatory regarding seasonality or role of copper. Thanks again for joining. I will request you to turn off your video